Welcome to this lecture on current methods in neurotechnology. I'm Ulrich Ettinger. I'm in the Department of Psychology at the University of Bonn, and I will now talk about human oculography. Just to give you an overview of the lecture, first I will say a few words about the scientific background, which is the evolution and classification of human eye movements. Then I will introduce the problem, which is how to unintrusively obtain precise and accurate recordings of eye movements with high spatial and temporal resolution. I will introduce some of the major techniques that are currently in use in human oculography. I'll give a short overview and I'll point out some of the advantages, limitations, and pitfalls of these methods. And then I will say a few words about data analysis, and I'll introduce some key concepts in oculography data analysis. So first of all, let me begin with a quote that is already quite old by William Porterfield from 1737, which I found in the book by Wade and Tatler. And Porterfield defines eye movements and distinguishes between internal and external eye movements. And it's really external eye movements that I will focus on today. So motions of the eyes are either external or internal, and external are those motions performed by its four straight and two oblique muscles. And it's those external eye movements of the eyeball relative to the orbit, which are controlled by the extraocular muscles, that I will focus on today. These are the eye muscles. There are 6 muscles that allow us to move the eyes rapidly and efficiently. There is the superior and inferior rectors controlling vertical eye movements. There are the lateral and medial rectors controlling horizontal eye movements. And then there's the superior and inferior oblique allowing rotational eye movements. A key architectural component or setup that we need to consider when we want to understand eye movements is the architecture of the eye, including the retina and its inside-out arrangement, with the photoreceptors here at the back of the retina and light traveling through the eye, reaching the posterior part of the eye, the retina, and the light has to travel through anterior cell layers and the axons of retinal ganglion cells before it reaches the photoreceptors. And this inside-out arrangement causes the light to be distorted before it reaches the photoreceptors. And the solution to this problem, which causes less than perfect acuity vision, is the fovea. The fovea is a very small part of the retina where the anterior and intermediate cell layers are pushed to the side so that light can reach photoreceptors uninterruptedly and without distortion, allowing highest acuity vision. And this is borne out in this figure here, where on the y-axis we can see the relative visual acuity. And as you can see, visual acuity is highest and considerably higher than anywhere else in the retina in the fovea. So visual processing is best when a stimulus, an object of interest, is viewed foveally. And this architecture is important to the evolution of eye movement in humans. In terms of the evolution, the first threat to clear vision, however, were head movements, for example, during locomotion. Without the ability to compensate or to use compensatory eye movements, vision would become blurred every time we move our heads or bodies. And a solution to this problem was the evolution of gaze stabilizing eye movements that allow clear vision during locomotion. There are vestibulo-ocular reflexes, very fast, rapid movements that compensate short and rapid body or head movements. And then there are visually mediated reflexes that compensate for prolonged, longer ongoing movements, the optokinetic and smooth pursuit tracking eye movements. A second requirement of eye movements that arose due to the evolution of foveal vision was the need to look at potentially interesting objects that may suddenly appear in the periphery so that they can be processed foveally. And so this is getting back to the architecture of the eye and the need to look at objects and details of objects that appear interesting, that need to be processed in further detail, to look at these, so to point the eyes at these objects so that reflection of light from these details or parts of objects is processed foveally. And the solution to this problem was the evolution of gaze shifting eye movements, particularly eye movements that are independent of conspicuous and resource demanding head or body movements. And so these processes led to what we now consider two main classes of eye movements, gaze stabilizing and gaze shifting. And we can categorize or classify eye movements along those lines. There is a more refined functional classification of eye movements by Lee and Z, which comprises the following: vestibular, fixations, optokinetic, smooth pursuit, quick phases of nystagmus, saccades, and vergence eye movements. And for reasons of time, I will only really focus very briefly on fixations, smooth pursuit, and saccadic eye movements. Fixations, first of all, are a widely reported type of eye movement, the most reported event in eye tracking studies, according to Holmquist et al. This becomes obvious from a number of psychological neuroscientific studies in humans and non-human primates, where the eye position or the gaze position is recorded while participants, while observers view natural stimuli, for example, while humans read, view scenes, and so on and so forth. So fixations can be defined as the resting of gaze on a stimulus, or again on a stimulus detail. And in natural viewing conditions, periods of fixation typically alternate with saccades. Now, the term fixation is something of a misnomer. It implies lack of any eye movement, but really fixation periods comprise considerable oculomotor activity. It's very small saccades, less than a degree in amplitude, termed microsaccades, or slow drifts, or high-frequency oscillations. And some of these eye movements can be seen here in a recording of someone viewing this particular image here, and then there's an enlarged section of essentially a fixation of this part of the image here, which shows saccades, the larger rapid eye movements, then the microsaccades, less than a degree in amplitude, and slow intersaccadic drift. Smooth pursuit are slow eye movements that serve to keep the image of a slowly moving stimulus on the fovea. Smooth pursuit eye movements can only be performed in the presence of a slowly moving stimulus. Pursuit really is the result of a complex interplay of perceptual, cognitive, and motor processes. Within the realm of perception, it's particularly emotional processing that plays an important role here. From the cognitive side, it's attentional and predictive processing that play a major role. Pursuit is mostly relatively slow. In laboratory studies, we typically use target velocities, stimulus velocities of up to 30 degrees per second, but pursuit eye movements can be made with velocities of up to about 90 degrees per second. We can measure pursuit in a number of different ways. There are both global and specific measures. Global measures include, for example, the root mean square error, and specific measures include primarily the velocity gain, that is the average eye velocity for a certain segment in time divided by the target velocity, or the frequency of saccades. And there are different subtypes of saccades that occur during pursuit: intrusive saccades, such as anticipatory saccades, or compensatory saccades, such as catch-up saccades. The neural network that underlies smooth pursuit eye movements in humans is quite well established. It's quite well known, mostly from human lesion and functional neuroimaging studies, but also from non-human primate studies. In this diagram from a chapter by Rebecca Lenzer and colleagues, we can see both the visual pathway from the eye via the lateral geniculate nucleus to the visual cortex, and then the oculomotor pathway emerging from visual cortex via motion sensitive areas MT and MST to oculomotor areas, eye fields in both the parietal cortex and in the frontal cortex, and then direct or indirect projections via the basal ganglia to pontine nuclei and via the cerebellum to oculomotor nuclei. And then finally, after fixation, smooth pursuit, I will now turn to saccades. These are rapid gaze shifting eye movements, jerk-like eye movements. The name Javal is closely associated with the term saccades, and saccades serve to bring the image of an object of interest onto the fovea. In everyday viewing conditions, we make about 3 saccades per second, and saccades typically alternate with periods of fixation. Saccades are really very rapid, with peak velocities of up to about 500 degrees per second, and they're very short movements. Durations are often only 20 to 50 milliseconds, and they're typically less than 100 milliseconds. A noteworthy feature about saccades is the very close law-like relationship between amplitude, saccadic amplitude in degrees, and the peak velocity in degrees per second, or the duration in milliseconds. And this relationship is termed the main sequence, and it can be seen here on the right in an image from a paper by Susana Martinez-Condo. Here are some illustrations of what saccadic or what recordings of saccadic eye movements look like. You can see here in the image, in that little movie on the right, the recordings of just one trial of an observer viewing the different options in an intertemporal choice task. And you can see the eye position, the gaze position on the screen, and you can see how fast these eye movements are and how short they are. And the same movements that you can see here on the right are shown here on the left in red horizontal and in green vertical eye movements over time. And the saccades have already been marked, detected by an algorithm. This is the data via software from SR Research. Saccades have been marked in yellow so that we can identify and then later obtain measurements about these. Like for smooth pursuit, the neural networks underlying saccades are already very well understood. The neural control in humans involves cortical eye fields, frontal and parietal cortex, the striatum, cerebellum, and brainstem areas. And again, much of this is based on evidence from functional neuroimaging studies in humans, but also from lesion studies and non-human primate studies. Now, with regards to the methods which we call oculography or eye tracking methods to record eye movements in humans. At present, there are a number of really quite well validated and often user-friendly methods that we can choose from. The most commonly used method in oculomotor research is what's called video-based combined pupil and corneal reflection tracking. There are other methods as well, however. Infrared Limbus oculography, there's the scleral search coil, which involves placing a contact lens on the eye, is therefore a little intrusive and not always well tolerated by participants, but by many researchers still considered to be a gold standard of human eye movement recordings. Then there's the EOG, electrooculography, dual Purkinje tracking, and then with great significance in neurological assessments, there are bedside assessments of oculomotor function. First of all, before I introduce some of these methods, some key terms in oculography. The first key term is precision. This is essentially the reproducibility of the eye tracker, of the device, of the eye tracker signal under identical conditions. So for example, if we used a glass eye and pointed it at a certain position on the screen, as it were, so the glass eye is looking at a certain position on the screen, then we would expect an eye tracker, a device with high precision, to give us the same signal again and again. And this is shown here on the figure on the right. There are 5 samples, which in precise recordings in the upper panel are closely clustered together. So on 5 different occasions and 5 samples, the eye tracker gives a very similar result. There's also something that we term accuracy, however, and this is also very important to consider. Accuracy is really the difference between actual and reported gaze position. And you can see that if the eye was really looking at this cross here in the middle of this circular array, then precise and accurate eye movements would, or recordings rather, would be reflected by the 5 samples clustering around the ideal viewing or gaze position. Precise and inaccurate would be shown up here, and imprecise recordings are then those with a considerable scatter. Another term that we need to be aware of is the resolution. There's both the temporal and the spatial resolution of eye trackers. The temporal resolution refers to the number of samples per unit of time. Typically, this is given in samples per second in hertz, and it is also known as the sampling rate of the eye tracker. With modern video-based systems, sampling rates can go easily up to 1,000 or even 2,000 hertz. The spatial resolution, on the other hand, is the smallest distance of 2 points that the system can resolve. Let me now introduce one of the main, if not the main method in human oculography at present. And this is the video-based combined pupil and corneal reflection tracking method. This is a very widespread approach in many contexts of eye movement research, in academic, basic science research, and clinical research, but also in applied settings, such as in the industry. This method provides a measure of the point of regard or point of gaze, that is the gaze position in space. On the screen, for example, the XY with a known Z coordinate, the XY coordinate that is being looked at by the observer. And this allows us then to identify which object or which part of an object on the display is being looked at, is being fixated by the observer. In contrast to other methods, 2 measures are drawn upon here. First, the corneal reflection of infrared light, that is the first reflection of light from the outer surface of the cornea, the first Purkinje image, and the pupil. And these 2 signals are combined to yield gaze position. The components of such a system include an infrared light source, an infrared sensor, a camera, a processor, and an algorithm to detect pupil and corneal reflection, or a 3D model of the eye. And different manufacturers use different approaches here. The light source and the camera, which is what we may call the actual eye tracker, can either be head-mounted, so worn on a band around the head like a bicycle helmet. It can be fixed to a chin or headrest, or it can be remote. That is, it can be placed below the monitor, for example. Here is an example of an illustration of pupil and corneal reflection images, the first Purkinje images. And as you can see in the center, this is an observer now looking straight at us. And as the observer makes horizontal, vertical, or diagonal eye movements, you can see the position of the pupil changing, whereas the position of the corneal reflection stays roughly in the same place. And this fact is used by this method to derive gaze position. You can see the first Purkinje image, the corneal reflection, when you closely look at someone's eye and light enters from the side, maybe from a window or from a bright lamp. You can actually see the reflection of light on the cornea. This is shown here in a paper in vision research. This is the first Purkinje image, or the so-called glint. And we can also then detect, this is from the SR Research eye-link eye tracker, we can then detect both the pupil and the corneal reflection with the crosshairs indicating the center of each of these signals. The method has been adapted in a number of contexts, for example, for mobile eye tracking. Here, the Tobii Pro glasses, which can be worn in traffic context, for example. That's one area of application, or in shopping or in usability context. And it can also be adapted or integrated into VR settings. The method has also been applied or adapted to fMRI. So in this setting, this is the SR Research eye-link. The participant lies on their back inside the MR scanner bore. The eye tracker, the infrared light source, and the camera are placed at the end of the bore, and the participant views the stimuli via a mirror which is placed on the head coil, and it is that same mirror that is used to send the infrared light, shine the infrared light onto the eyes of the participants, and to pick up the reflection for eye tracking. Now, some of the advantages of this method are its high accuracy, its excellent resolution, temporal resolution, up to 2000 Hertz. It's relatively easy to use. It provides not only measures of gaze position, but typically also of pupil size, because the pupil is tracked as one of the 2 signals in this method. Some of the disadvantages include that there may be artifacts due to eye makeup, and then there's something called post-saccadic oscillations, and these are really due to pupil movements relative to the iris. This has been shown in a paper by Nyström and colleagues, which I show you here, in recordings from video-based combined pupil and corneal reflection trackers, after saccades, at the end of a saccade, you sometimes see these oscillations. This is a single participant here making horizontal eye movements. And when this was studied closely by Nystrom and colleagues, it was found that it is more likely the pupil center that makes these oscillations rather than the center of the iris. So it's essentially, at the end of the saccade, the pupil wobbling inside the iris. So these are not necessarily true eye movements as such, or external movements as Porterfield would say, but rather internal movements. And so we have to interpret these accordingly. This is an artifact that you don't get with other methods, for example, with the infrared Limbus tracker. Infrared Limbus trackers are based on the anatomical feature of the eye, that is the Limbus. The Limbus is the border between the sclera and the iris. The sclera is the white of the eye, and the iris is the colored bit of the eye. And here, infrared light is shone onto the eye from a relatively short distance, typically from a head display, a display that is worn, a device that is worn on the head. So the infrared light is shone directly onto the eye and picked up again by photodiodes. It is known that the iris and the pupil as well reflect less infrared light than the sclera. And this difference in intensity of reflected light can be used to derive eye position or sort of the position of iris and pupil, and therefore to allow measures of eye movements. Advantages include the good temporal and spatial resolution, no contact lenses are necessary, and changes in pupil size, which do of course occur in cognitive and visual experiments, do not affect measurements. Disadvantages include the relatively low recording range, especially for vertical movements. In this setup, the light emitters and detectors alone would reduce the range in the vertical direction. Susceptibility to artifacts, for example, from head movements or sliding sensors. And finally, the head set can be a little uncomfortable, especially over longer periods of time, if it has to be fixed relatively tightly to the head. Now, finally, we would then like to look at the data that we've obtained from our recordings. Oculographic data, in its simplest form, are a stream of X and Y coordinates of eye position over time at a given sampling frequency. And this is for a standard setup, where the Z coordinate, as it were, the distance from eye to the monitor, to the display, doesn't change. So we're then interested in simply horizontal and vertical and diagonal movements on the monitor. And the first step in data analysis is often the detection of what we call oculomotor events. Events are saccades, for example, or segments of fixation, or segments of pursuit. And these events, these different kinds of eye movements, are typically identified on the basis of certain criteria. So we need to simply apply criteria to identify these in the stream of data. And these criteria could be durations. We would like saccades, for example, to have a maximal duration of 100 milliseconds, or a minimum velocity criterion, a minimum acceleration criterion, and so on and so forth. To give an example, some example criteria for event detection for saccades, these short, rapid eye movements, we may wish to apply a minimum, or typically apply a minimum velocity criterion of, let's say, about 30 degrees per second, a minimum acceleration criterion, and also typically minimum amplitude criteria, let's say about 1 degree of amplitude, to differentiate saccades from microsaccades, for example. Then, because saccades typically alternate with fixations, we may want to be sure that we can successfully separate saccades from fixations. So for fixations, we would say, well, these are gaze position, or these are segments where gaze position falls within a certain spatial area with a maximal amplitude of 1 degree. So we allow for some drift, but only minor drift and not major movements. And also we apply a maximal velocity criterion and perhaps a minimum duration. To give you an example of what this may look like, here again from the book by Holmquist, Kenneth Holmquist and colleagues, on the y-axis, the velocity, on the x-axis, the time. These are data from someone reading a couple of lines of text. You can see the fixations, the little nodes here, and the saccades, the lines in between. And here someone was, the observer was fixating for a slightly longer time period on these letters. And so for a dispersion, as a dispersion threshold, we would say that, well, to qualify as a fixation, the data samples must fall within the circle, and there could be a 1 degree amplitude circle for a certain amount of time. And then we would classify that as a fixation. For the velocity criterion, we could say that, well, 30 degrees per second is our cutoff. So samples that are below 30 degrees per second and have a certain dispersion and maximum dispersion and minimum duration are then fixations, and anything above are then saccades. What can we then measure about these different types of eye movements once we've identified them in the stream of data? Well, the answer is we can measure a lot. That's one of the nice features of human oculography in general, that in relatively short periods of time with a high temporal resolution, we obtain a lot of data, and we can then obtain a lot of different measures. We can derive a lot of different measures from these data. There's a taxonomy of parameters by Holmquist and colleagues. These relate to movement, position, or these describe movement, position, numerosity or frequency, and latency. For smooth pursuit eye movements, for example, we could first of all consider the eye velocity as a movement parameter. So for a segment of pursuit, this is a constant velocity target here in red, moving from the left to the right. Over time, we could say that, well, for the middle 50% of that ramp, we would like to calculate the velocity of the eye, the average velocity, and divide it by the average velocity of the target to derive the velocity gain. We could also then count the frequency of saccades that occur during the entire task. For example, there are a couple of saccades here, there's a third saccade there that we can literally see, and we can apply our criteria to identify that, to be sure that these are really saccades. For saccades in a saccadic task, in a saccade paradigm, we could then derive further measures from saccades. So here's an example of a target that steps or moves abruptly from one position to another horizontally, and the eye of the participant follows the target as it were. So a rapid eye movement, a saccade, is made with a spatial correction sometime afterward, and then within this eye movement, from this saccadic eye movement, we can derive a number of measures. For example, the amplitude. This is the data viewer surface from SR research. From this saccade, we can now obtain its amplitude, its peak velocity, its average velocity, and so on and so forth. Now, you may ask, why would we do eye movement research? What are the possible uses? What are the what are some of the contexts that eye movement research have been, or eye movements have been studied in? There are a number of contexts, many more than I can talk about in this brief lecture, but I'll give you just 3 examples. The first example is basic science, basic cognitive neuroscience. These are 2 illustrations showing, or 2 panels showing recordings from a non-human primate brain and the bold signal from fMRI of a human, both during a saccadic eye movement task, specifically a memory-guided saccade task. So in this memory-guided saccade task, a cue, a stimulus, is shown in the periphery while the participant fixates, looks at the center of the screen. And then after a short period of time, a saccade has to be made to that cue. The cue has since disappeared, so the position of the cue has to be held in visual spatial working memory. And this is a very popular task, the memory-guided saccade task, to probe visual spatial working memory in humans, but also non-human primates. And Patricia Goldman-Rakich used the task to describe and to help understand the role of the prefrontal cortex in visual spatial, in enabling visual spatial working memory. So here, a saccadic eye movement task was essentially used as a tool to study the neural mechanisms, the neural mechanisms of visual spatial working memory. A second example is the use of eye movement measures as biomarkers. And there's a considerable literature, there's a considerable interest in industry in using eye movement tasks and deriving eye movement measures to study drug effects. The saccadic peak velocity, that is the maximum velocity that the eye reaches during a saccade, is known to be highly sensitive to sedative agents, benzodiazepines, for example. It's also very sensitive to psychological or physiological effects like fatigue, time on task, and so on and so forth, even motivational influences. And so these are, this is an example taken from the Center for Human Drug Research in the Netherlands, one of a number of industry partners or companies that work with saccadic eye movement recordings to study drug effects as or to identify sedative effects of drugs. And so what is shown here in this little example is that the saccadic peak velocity in the range of 400-500 degrees per second is reduced quite considerably by a sedative agent relative to placebo. There's also an effect of time. So over time, peak velocity tends to reduce when we make saccades repeatedly. And then a final example. This is now a very different context, again, where eye movements are recorded and data derived from eye movement recordings are used in an applied research setting, and this is web usability research. So here you can see the gaze position, the fixation data of someone viewing this website, and this is what we call a heat map with cold colors, blue indicating fewer or shorter fixations, and warmer colors, yellow, orange, and red indicating more or longer or more frequent fixations. So using that approach, we can identify where on a given website someone is directing their overt attention, someone is directing their gaze. Now, with these three examples of eye movement research in basic and applied settings, I'd like to conclude. I'll summarize what I've said. There is a broad repertoire of human eye movements. This has evolved to allow both gaze shifts and to allow stabilization of gaze. The neural mechanisms of eye movements are already quite well characterized from studies of humans using lesion and imaging methods, but also from non-human animals. Oculography, eye tracking, can provide a rich amount of data on eye movements with excellent accuracy, precision, and temporal and spatial resolution. These methods, some of these methods have been adapted to allow assessments not only in the oculomotor laboratory, but also in fMRI and in real life using mobile eye tracking. And there are a number of applications of eye movement research, from usability to biomarker research in the pharmaceutical industry, over and above basic cognitive neuroscience research. These are some recommendations on further literature. There are a number of books, some of which have been published in the last years, on eye movements, on eye tracking, oculography as a method. And with that, I also give you some website to consider, some eye movement related websites that may be of interest to you. And with that, I conclude. I thank you for your attention, and I'll leave it at that. I stop the presentation. Thank you.